Thank you. It's not only the schedule change, it's also topic change. Because I thought it's late in the day, you're tired, and what I was planning to talk first is long-read genome assembly, which is a little bit heavy. So I thought we will kind of make it lighter today, and we will also have fun because we will be playing a game, and it's also more relevant to metagenomics talk. Okay? Agreed on? Good. This is Larry Smarr, who is a professor in my department, computer science professor, who is probably the most well medically studied man in the world. He turned himself into a walking laboratory when he was diagnosed with the Crohn's disease 10 years ago, and he meticulously collected stool samples with detailed metadata description of what antibiotics he took, how he felt. So it's enormous and very useful set of data that we have access to. What is amazing about this data, it's the largest metagenomics time series ever generated, and it's also metabolomics time series. That is rare. So it's a paired metagenomics and metabolomics data set. So and I work both in metagenomics and antibiotic discovery. So when at some point we found exotic flux cyclic peptide with antibiotics activities in Larry's stool, I thought our program doesn't work. And then I called Larry, and it turned out, of course, he eats flax-seeds because they're apparently beneficial for health. His diet is very advanced and controlled, which means that basically, and these flax seeds, as we demonstrated, survive the entire gastroenterological tract, and we can match the dates when he ate flax seeds to our samples. So interesting question is, when we eat plants that have compounds with known antimicrobial activities, we're essentially exposing ourselves constantly to antibiotics. How these antibiotics affect human microbiota remains to be seen, but we definitely can see a lot of antibiotics from plants in human microbiota. And this is Larry with Rob Knight, who actually sequenced his metagenomics time series. Every point here is Larry's data point in microbiome, and Rob explains Larry what's going on here. But you don't have to explain much to Larry because he turned himself into pretty advanced biological expert after he started this metagenomics time series studies. And this is Sergey Note from my lab, who has actually assembled all this metagenomics time series, many data sets. And you heard from Ryan how difficult it is to assemble a single data set. Single large data set. So last year, Sergey used up 1 million hours running Metaspace that Ryan was talking about at San Diego Supercomputing Center, assembling Larry's data sets. Okay, so these are, and Ryan already talked about Metaspace, and Metaspace is just a part of efforts to turn Space assembler, that is now the most cited assembler, into a universal assembler, because there are not a single assembly task, there are dozens of different assembly tasks. And we have now about 12 different Space tools from Space family. So Metaspace, that's what Ryan was talking about. Meta time series, that's something that we are about to submit, also developed by Sergey Note. Also hybrid space that we are using for metagenomics assembly of long and short read in the hope to extract complete genomes from metagenomics assembly. Metaplasmid space to reveal enormous variety of plasmids in existing data sets that are currently remain below the radar of existing techniques. And Ryan was mentioning some Mong data sets that are well-studied artificial data sets. We were amazed to find still unknown plasmids even in Mong data sets. And finally, true space, that is also a way to develop very accurate metagenomics assemblies that go beyond the limits of existing short reads. Okay, who works on human microbiome assembly? Please raise your hands. Very good. So how many different species you expect to find in a healthy human microbiome? How many? I don't need exact number, roughly. Definitely not. If you find 100, it means that the person is seriously sick. So Larry is interested in this project particularly because he is constantly exposed to antibiotics. And diversity of microbiome is a very important characteristic of the health of microbiome. He wants to know how diverse his microbiome is. So healthy human, it's actually not trivial to estimate the number of species in the human microbiome.
It also depends on the depth of sequencing.
No, it doesn't depend on the depth of the sequencing, because I'm asking general biological questions that doesn't depend on the technology. I'm not asking how many you see. I'm asking how many players are there. And usual consensus, maybe 1,000, roughly 1,000 in healthy human microbiome, roughly 1,000 species. Next question to people who assemble actually microbiome. How many species you see in a usual assembly? What? You are extremely lucky if you see 400 or 500. De novo shotgun assembly. I'm talking about de novo shotgun assembly. I never saw de novo shotgun assembly with 400 or 500 species. Usually, in our hands, maybe 100, 200. So we are seeing 10, maybe 20% if we are lucky of what is there. How can we find how complex is actually human microbiome? Okay? So now imagine this scenario. Rob Knight generated 50 million of reads. Sergey Note constructed assembly with total length 250, 60 megabase. So these are all contigs ordered in increasing order of coverage. And this is the curve. I arrange all contigs here that abruptly stops at 260 megabase because you cannot assemble much when the coverage drops below 5x, right? That's what we see. So both Rob and Sergey look happy, but Larry is not happy. He wants 500 million reads. And maybe with 500, to increase the number of reads by a factor of 10, and maybe with this increase in the number of reads, he will see much more of his genome. Will he? Nobody knows, right? Maybe it will be a waste of time and effort. So the question is, can we, in the traditional metagenomics assembly, access the rare biosphere? Okay? And rare biosphere means 90% of our microbiome. And currently, metagenomics assembly is limited to this top 10% of species. And rare biosphere is very important. It's antibiotics production, something that I'm very interested in. It's infectious agents, it might be viral, and it's adaptation of the environment. Okay? And therefore, the goal that I'm trying to address, this is, if we would continue this curve, it will go far away. Usually beyond, usually it would go to 3 billion. Okay, but for this particular case, let's stop here. And this is hidden microbiome. We have no access to hidden microbiome unless we dramatically increase coverage and makes it very expensive or go to new technology. But can we at least estimate hidden microbiome? Because that would give us information about diversity. And diversity is linked to disease, In human, in corals, limited diversity, sometimes excessive diversity also leads, is not a good sign. So this is the goal. Is it clear? Do you hear me in the end? Okay, good. Sure. I will be, so far I talked about short-read technology, but a little bit later I will talk about other technologies. I'm doing whole metagenome. I'm doing shotgun metagenome sequencing. Let's talk about diversity. There was a shocking paper 2 years ago by Lucy Lennon that claimed that there are 1 trillion of microbial species on Earth. Currently one of the most cited papers in PNAS. And amazingly, in the same months, there was a paper from Schloss claiming that there is only 1 million of bacterial species on Earth because extraction of 16S RNA approaches saturation. This illustrates how little we know about diversity. And this is about diversity on Earth, but we are interested in a more tractable question. What is the diversity of a given sample? How many species live here? And in this case, estimates also vary widely. We actually have, when we sequence metagenome, we don't have a good estimate of diversity. We only see what we can assemble. Now, there were thousands and thousands of studies that try to estimate diversity. It's a classical topic. Many of them, most of them are based, basically all of them are based on 2 approaches. It's either parametric estimators that assume that genomic abundances follow a known distribution. And there is a lot of criticism of these approaches because you never know what is the parametric distribution. Adequately model a sample tail is notoriously difficult to approximate, and parameter estimation often rely on 16S, which is unreliable. And another is non-parametric estimators that assume that there is a good database of known species. And everybody who worked even with human microbiomes, the most studied microbiome, knows that there is no such database yet. And that's why we propose a new approach to diversity estimation. No assumption, no parametric assumption, do not require a database, account for rare species with read coverage as low as 0.01x, something that's completely invisible for assemblers.
Before you move on, what definition of species are you using?
We'll come to this in the last slide of my talk. This is, of course, a difficult question, but I will answer it.
I was just wondering functionally how you consider it.
Functionally, nobody will be able to answer this question, but I will be able to answer this question computationally. I will give like computational definition for how we treat similar species. Okay. Okay. So what we will be doing, we will be using some strange arguments from geometric probability to analyze both short and long reads. I'm arguing that if you have only short read, it's very difficult to estimate diversity. If you have only long reads, it's equally difficult to estimate diversity. But when you have both, there are amazing mathematical possibility that is not immediately obvious that you can actually estimate diversity. And there are 3 technologies. And for the sake of simplicity, I will talk about this synthetic long read technology, simply because it's a little bit easier to handle for my goals. And who knows about synthetic long read technologies? Some people know, good. So this is a technology that generates roughly 10 kb long, very accurate reads, pseudo-reads, virtual reads. The reads that are 10 times more accurate than Illumina reads. And it is a technology similar to 10x technology, but it is 10x technology with high coverage of every fragment. In 10x, coverage of each fragment is very small, light. In true synthetic concretes, it's high. And so the technology works like this. DNA is shred into 10 kb long fragments. Fragments are distributed among 96 pools. Pools are barcoded in sequencing, and short reads are assembled into synthetic concretes. If you don't understand this, not a problem. The only thing you need to realize is that there is a way to generate synthetic concrete that are accurate and long. It can be packed by reads, doesn't matter. I just don't want to get you into difficulties of dealing with packed by reads and mapping of Illumina reads against packed by read, because Ben knows how difficult it is sometimes. Okay, next. Next, we'll talk about something completely mathematical. This is my metagenome. Metagenome consisting of 6 species. Metagenome richness is defined as the total number of distinct genomes in the metagenome. And metagenome length is defined as the total length of all genomes within metagenome. Is it clear? Okay. Now, for each genome within metagenome, we will construct a genomic rectangle. The base is the length, and the height is the number of cells of given genome in the population. Huge, huge rectangle, right? Because there are probably millions of cells. What? There is no coverage. I'm talking about metagenomic sample. In this sample, there is no technology here, only biology. In this metagenomic sample, I have 1,000 cells of this organism. And I simply build this rectangle with width equal to the length of the genome and height equal to 1,000, the number of cells. Okay? It's just abstract notion. And yes? I haven't decided yet. I just consider abstract problem. I have collection of n genomes, and I'm an oracle. I know how many cells of each genomes are there. And as an oracle, I can construct this rectangle, right? I'm not claiming that I know these numbers. I know this number maybe, but I'm not claiming that I know this number. I just currently thinking about an abstract problem, and then we'll return back. Is it clear? And metagenome is represented as a set of rectangle, and total area is sum of areas of this rectangle, and metagenome area is the total length of all DNA extracted from metagenome. I think it's pretty clear, right? Okay. Where do we sample reads from? We sample reads from this area, right? Let's assume we uniformly sample reads from this area. And this rectangle diagram are very large. Let's normalize it by the area. So the total area will be 1. So the width of each rectangle equal to genome length. The height of each rectangle is normalized over the total area. So we divide everything by total area. An area of each rectangle is normalized as simply area divided by total area. So abundance is now is a fraction of area among all rectangles. Make sense? Okay.
Do you mean abundance of the reads, or you mean abundance of
No reads yet. There is no reads here.
the number of microbes should be the height, right?
Number of microbes is for every number of cells for each species is the height, but I normalized everything by a huge number. That is the total area of all rectangles. And now the height,
Of all rectangles.
yes, of all rectangles. So I normalize everything. So it's now very long and height is very small, right? Okay. Now, what is a probability that a read originates from a given genome? Of course, it's abundance. It's fraction of area of this genome among all areas, and all areas is 1. And probability is that a read originates at a given position of this genome, right? We simply need to divide abundance by the length, which is exactly height of the genome. And that's how rectangle diagram looks like for a real mock metagenome, probably one of the metagenomes that Ryan was talking about. And this is in decreasing coverage. And if we start counting cumulative abundance, then we can introduce a term similar to NG50, which will be, I call it M50, which is what is the total length of all genome that account for half of the length of metagenome. And in this case, it's very simple. Mock metagenome, 20 species. M50 is 7 megabases. Okay. Now, let's play a game. So, and of course, there will be Alice and Bob. So here's the game. Purely mathematical game, has nothing to do with metagenome. Alice has a set of rectangle with total area 1. Okay? And each rectangle is characterized by length and height. Bob doesn't know the number of rectangles and the length or height, nothing. Okay? And his goal is to estimate the total rectangle length by asking n questions. Total rectangle length is the sum of lengths of all metagenome. It will provide us roughly with the number of species if we assume that we know average genome length. Okay? So what questions Bob can ask? Bob can randomly sample a point from the area, and Alice tells Bob what is the height for this point. Okay? And Bob has n questions to ask, and then in the end, after this question, Bob has to answer what is the total rectangle length. Make sense? Give me a break. How possibly Bob asking question about the height and knowing nothing about the length can estimate the length of metagenome? How is it even possible? Alice doesn't provide any clue about the length, only about the height. Okay, good. So let's see about how uniformity helps. No, of course, Bob asks uniform question. You get ideas about height, and then what? What do you do with this height? You have 1,000 of estimates of heights. How do you derive length?
Maybe you can improve the area size.
Area size is 1. Always.
Yeah, but like based on what you have. So you say, well, say I have a point at a given length and for given height, so you can kind of assume the area that's already taken and the area that you kind of have. You should have to get 1 in total.
But Bob has no clue from what side. Every point is sampled from certain height. Bob has no clue where, how long. Bob has no height about this coordinate. Only heights. Anyway. And how does it help? You construct distribution of height. How does it help? You have height distribution, but heights are distributed over the lengths. How do you know the lengths? We are interested in distributing, right?
There are more chances that you'll get a point in a wide rectangle than in a narrow rectangle.
Good point. So it's simple. Let me show you how it's what is happening. So total length of all metagenome is simply sum of lengths of all genomes within metagenome, which is length of every rectangle is area of rectangle divided by its height, right? Area of rectangle is height by length, So sum of length is simply sum of area divided by height, which is, I will substitute area simply integral through this area. And as a result, I simply have an integral over the whole metagenome area, the whole rectangle length. And total length is essentially integral over the whole area of inverse height. So this is the key point. Bob has to look at inverse height to get an idea about the result, and therefore the only thing Bob has to do is to plug estimates of heights into this formula to get the length. Make sense? Okay. And moreover, using similarly a little bit more involved arguments, Bob actually can reconstruct the whole rectangle diagram. Not only the total length, but the whole rectangle diagram, if there is enough questions then.
Pavel, so he has n questions?
Yes.
N questions, and n is the number of species.
No, n is a parameter. It may be much larger than number of.
It must be much larger. How much larger?
We'll see it in the next few slides. There is no, we are talking about abstract problem at the moment. We'll come. It's a good point to show the next slide.
No, not even practical, but even theoretical. How much larger than number of species
in order to accurately estimate?
Yeah.
This is a good question that I will try to answer a little bit later, but this is perfect question for the next slide. Here's the next slide. Biologists are falling asleep because it's absolutely not clear what this game has to do with DNA sequencing, right? So that's what it has to do with DNA sequencing. So Bob generates n long reads. N is a parameter. So typically it will be, let's say, 100,000 usual number, much larger than the number of species in the genome. And they uniformly sample the rectangle diagram. Bob doesn't know anything about the shape of the diagram, rectangle diagram. Alice, on the other hand, generates k short reads and maps them to each long read. Alice has to do mapping, and that's why I didn't want you to present it with error-prone Pacific Bioscience Oxford nanopolar read, because mapping becomes tricky. Not impossible, but tricky. And coverage of a long read by short reads provides the height estimate of every rectangle in metagenome rectangle. So Bob generates long reads that sample rectangle diagrams. This is each red line is long read. And Alice basically responds with the number of how many short reads map to this long read. For example, for this long read, single long read here, there are 22 short reads. It's the parameter number of reads. And the mini rectangle with this base, length of this long read, has area number of reads over k, because we assume that short reads are uniformly sampled from the rectangle. And height, then, number of reads divided by k and divided by length of the read. And this is another read that provides a different estimate for the number. And then our assumption is n long and k short reads uniformly sample the metagenome. Short reads can be accurately aligned to long reads. And if this assumption holds, then the total length of metagenome is simply for every, there are n long reads, there are k short reads. For every long reads, we estimate, we know its length, and divide by the number of short reads mapping to this long reads. And this is the estimate for the total length of metagenome. I'm hiding many details here, right? And one of the important details is that we have unbiased estimator for the height, but we are actually need to imply to find unbiased estimation of the inverse height. And this results in some challenges that, however, let me just briefly run through this because I don't want to get into too many technical details. But if we look at the error in the inverse height estimators, it's not too bad. So we use this in our improved calculus. Even more important issue is there are repeats in the genome, right? And this repeats biased estimate of coverage because repeats from unrelated region may be gravitating to the same read. But our reads are short. We assume that most repeats are actually not as long as long read. And therefore, when we map reads to long reads, what we do is, like, there is a part of long read here that is extremely well covered, right? Maybe it's repeat in this area, and that's why it is so well covered. And there is a part that poorly covered, which is also seems to be abnormal. Maybe it's something related to GC composition. So we remove, for every read, we break it into sub pieces, sub fragments, and remove abnormal fragments from the estimate of the coverage to reduce the influence of repeats. And now, going back to your question about what is a strain, we can debate till the end of the day what is it, but for us, the question is, suppose we have two strains with genomes different by 1%. Should we count it as a single genome or as two genomes? Well, what would you suggest? What? Yes, it says, but it doesn't provide a solution. How do we deal with this? It's important to realize that every two cells actually differ from each other, right?
I don't know if the percent difference across the entire genome is very meaningful. As you go across the tree, there are some genes that because you're counting every position equally.